Hey guys, welcome to another lecture on Gametogenesis part 2, and today we're going to be discussing spermatogenesis. Right away, I'm going to first tell you the major differences between oogenesis and spermatogenesis because that can be asked as an SAQ. Firstly, spermatogenesis has no prenatal phase, which means that it begins at puberty. Also, unlike oogenesis, which ends at menopause, spermatogenesis has been shown to continue throughout life. Secondly, the entire cycle of spermatogenesis takes about 74 days. Okay, so it's not as long as oogenesis, which could take about even 40 years for one primary oocyte to be stuck in its prophase, like probably this lady over here. Next, remember that one primary oocyte eventually gives rise to only one ovum and two polar bodies, while one primary spermatocyte gives rise to two secondary spermatocytes, which eventually gives rise to four spermatids. Okay, that's an important point of distinction. Next, the cyto-differentiation. This is a typical feature that occurs in spermatogenesis, where a spermatid is eventually converted into a spermatozoon, where there is no mitotic or meiotic division, but just a metamorphosis and a rearrangement of the cellular organelles. Finally, the cytoplasmic division is equal in case of spermatogenesis. You saw in oogenesis how it is unequal, giving rise to a small tiny polar body and most cytoplasm going to the oocyte, secondary oocyte. Well, in this case, the cytoplasmic division is equal. However, the last stage of mitosis, which is known as cytokinesis, is not exactly completed in spermatogenesis. So for quite some time, the entire cells that are born out of a parent spermatogonium stay connected to each other by what is known as cytoplasmic bridges. It is only towards the end stage of spermatogenesis that these cytoplasmic bridges are eaten up by the Sertoli cells, so that the spermatids eventually leave, enter the lumen, and carry their journey forward. Okay? So, with these points in mind, let us talk about spermatogenesis. Spermatogenesis can be defined as the process of development of a primordial germ cell into a spermatozoon, and it occurs via mitosis, meiosis, as well as cyto-differentiation. Now, the mitosis and the meiosis can be considered into a process that is known as spermatocytosis. Spermatocytosis. While the cyto-differentiation or the metamorphosis can be defined by spermiogenesis. Okay, spermiogenesis. It's also known as spermatoteleiosis. So let's have a look at the various stages of spermatocytosis first. So in spermatocytosis, firstly at birth you have the primordial germ cell, which by puberty is converted into what is known as a spermatogonium. There are various types. There is type A spermatogonium, which is further classified into type A dark and type A light, out of which the type A light further becomes type B spermatogonium, and the type B spermatogonium goes through the further stages of primary spermatocyte, secondary spermatocyte, and spermatid. So let's have a detailed look at the process of spermatocytosis. But before that, let's go back to the developing testis around birth. So, you know that the primordial germ cells are special cells that escape the primitive streak, and by about the end of 5th week, they are settling themselves in the developing gonad, in this case the testes. Here, they form what are known as sex cords. And the sex cords, a typical sex cord will look something like this. It has the primordial germ cells and some accessory cells known as Sertoli cells. Now, by the time puberty strikes, these sex cords get canalized and form what are known as seminiferous tubules. Now, you'll study that more in detail in histology, and it's very important in histology. But for now, let's not get into details. The seminiferous tubules, after taking a section, will show two types of cells. One are the spermatocytic series of cells, and the other are these yellow cells that form a huge structure with extending cytoplasmic processes everywhere, and this entire yellow cell is known as a Sertoli cell. Okay, so you have the spermatocyte series as well as the Sertoli cells. Other than that, you also have in between these seminiferous tubules a special type of cell that is known as interstitial cells of Leydig. Okay, now it is the function of these interstitial cells of Leydig to form testosterone. Okay, they are equivalent to the theca interna, if you remember, which secretes estrogen. Okay, so testosterone is secreted by the interstitial cells of Leydig, while under the effect of FSH, the Sertoli cells start developing androgen receptors. Okay, so the testosterone secreted by the interstitial cells of Leydig is captured by the receptors of the Sertoli cells, and these Sertoli cells eventually protect and nourish and help in the development of the spermatocytic cells through its various stages of progression. Okay, also remember that the interstitial cells of Leydig are stimulated by the same hormone we studied in gametogenesis, that is luteinizing hormone, except in males it is called interstitial cell stimulating hormone. Okay, so that kind of takes care of the hormonal regulation of spermatogenesis. Now let's have a look at spermatocytosis. So as you know, the primordial germ cells give rise to what is known as a spermatogonium, and the spermatogonium that we study first is known as the type A spermatogonium. There's a couple of consequent mitoses that happens till we reach here, and then the type A can further be divided into type A dark, as I mentioned before, and type A light. Just remember that the dark appearance is because of the fact that the chromosomes inside are folded and neatly arranged, which means this is not an actively replicating cell, and it starts becoming light because the chromosomes start unfolding, which means that it's an actively replicating cell. And eventually every light type A spermatogonium gives a dark and a light cell. So the dark cell stays in the stem cell reserve, while the light cells, or the type A light cell, further and after subsequent mitosis becomes a type B spermatogonium. This again further undergoes mitosis to become a primary spermatocyte. Now remember that from primary to secondary spermatocyte is where the first meiotic division takes place. And remember that this is slightly prolonged. Of course, not as prolonged as the oocyte, but it takes about 22 days because of the long prophase of the primary spermatocyte. Now you know how prophase is long and it's divided into 5 stages, right? So finally, after the long prophase and after the long meiosis 1, we reach the secondary spermatocyte stage. Here the diploid content has become haploid. Please remember that. So one primary spermatocyte has given rise to 2 secondary spermatocytes, both are haploid, and these further give rise to 2 each. So now you've got 4 haploid spermatids. Okay, try to understand that everything that I've just explained is still not completely undergone cytokinesis. So all these cells are kind of going to be stuck to each other via cytoplasmic bridges. Now these 4 haploid spermatids are going to just, without multiplying, differentiate into what are known as spermatozoons, and this process is known as Spermiogenesis. Okay. So what exactly constitutes spermiogenesis? Well, let's take one organelle at a time. Firstly, the nucleus becomes super condensed and compact and becomes a little more oblong and spear shaped. Secondly, the Golgi body starts forming multiple vesicles or vacuoles, and all of them coalesce to form one big vacuole that surrounds the spear-shaped nucleus, and this is known as the acrosomal cap, and it has all these proteolytic enzymes which we shall study during fertilization. Thirdly, the mitochondria gather together and accumulate into the central area just below the head, forming what is known as the middle piece. They arrange themselves spirally over here. And the region between the head and the middle piece is a slight constriction that is known as the neck. And in the neck, you have one of the paired centrioles. So the paired centrioles essentially undergo multiplication. One is the proximal pair of centriole that lies here and gives rise to the 9+2 microtubule arrangement of what is known as the principal piece. And the distal centrioles just form a ring-like structure known as the annulus, through which the principal piece goes through, right? So that's your proximal and your distal centriole. And finally, the plasma membrane just surrounds all of these things, and whatever the excess cytoplasm is extruded. Rather, it is phagocytosed by the Sertoli cells. So we've seen that the Sertoli cells are helping in the nourishment, in the progression to various further stages of spermatocytosis, as well as the phagocytosis of the extra cytoplasm, which is also known as residual bodies. Right? The cytoplasmic processes of the Sertoli cells also help in forming an antigenic barrier, but we are not going to discuss that today. If you can find that out, it would be very interesting. An antigenic barrier, to be explained more in the histology. Okay, great. So the process of spermiogenesis essentially includes condensation of nucleus, coalescing of the Golgi bodies to form the acrosomal cap, mitochondria arranging themselves in the middle piece helically, proximal and distal centriole arranging to the proximal and distal part of the middle piece, and the microtubules finally forming the tail in a typical 9+2 arrangement. Okay, great. Now, spermatozoon structure and spermatozoon journey. You already saw the spermatozoon structure before. What you see over here in light blue is the plasma membrane. The dark pink is the acrosomal cap. The green is the nucleus. In purple, you've got these helically arranged mitochondria. You've got the proximal centriole at the neck, the distal annulus over here, and you can see the 9+2 arrangement of the filaments, microtubule filaments, as they form the tail. Okay, so we've got a head, we've got a neck, we've got a middle piece, and we've got a tail. Okay? Now the journey of the spermatozoon is also very interesting, because as soon as the spermatozoon is formed, it directly enters the lumen of the seminiferous tubules, but it hasn't really acquired complete motility. So it is the contraction of the seminiferous tubules that pushes these spermatozoons towards the epididymis, where they are stored for some time. Here they are taught a few more things. There is a certain exchange of glycoproteins in its plasma membrane, because of which it finally attains motility. So that's an important MCQ. The spermatozoons attain motility in the epididymis. And once they attain motility and finally during ejaculation reach the female genital tract, another important step actually happens in the female genital tract, and that is known as capacitation, which we shall study again in fertilization. But just remember that it happens in the female genital tract, either in the uterus or the uterine tube, and that is where some more exchange takes place in terms of glycoprotein molecules on the cell surface. Okay? Great. So, if we were to summarize spermatogenesis, the primordial germ cells at birth eventually by puberty get converted into spermatogonia. The type A dark spermatogonia will give rise to a pale as well as a dark spermatogonia. The dark one stays in the stem cell reserve, while the pale one further gives rise after subsequent mitosis to a type B spermatogonium. Each type B spermatogonium further mitoses into a primary spermatocyte. This is the stage where the meiosis 1 happens and diploidy is now becoming haploid to form a secondary spermatocyte. A secondary spermatocyte eventually forms the second meiotic division, which is just mitosis, to form spermatids. Correct? Now these spermatids are already showing some changes, as you can see over here, and eventually the excess cytoplasm is lost and the spear-shaped spermatozoon is created by spermiogenesis. These are your residual bodies, while the spermatozoa are free to go from the lumen of the seminiferous tubule all the way outside the male genital tract to enter the female genital tract at coitus. Okay, the entire process takes about 74 days, out of which the prophase is the longest, the prophase of meiosis 1, which lasts for about 22 days. Alright, remember, the spermatozoons achieve motility in the epididymis and learn the process of capacitation in the female genital tract. Okay? So, spermatogenesis is thus over, and we studied the mitosis, the meiosis, as well as the cyto-differentiation aspect of it. Now, it is up to you to find out the clinical aspects such as abnormal sperms and what is a normal and abnormal sperm counts, etc. So, that's going to be your assignment. Thank you so much for listening. Now that we know about the ovum and the sperm, let's discuss in the next session about both of them getting into holy matrimony and leading to fertilization. Thanks for listening. Bye-bye.